right. Hi, everyone. We're going to start a chapter on immunology, or acquired or adaptive immunology. Um, I just wanted to mention regarding this slide and this picture. This is actually the Russian army who forcibly vaccinated people against smallpox. Um, they used a live vaccine, of course, which is no longer used, right? And it killed between 30 and 50% of the people who were vaccinated with it. So for a long time in Russia, the older generation warned the younger generation against vaccinations, told them not to be vaccinated. Of course, today we use much safer vaccines. They're either attenuated or they are acellular, or in the case of the vaccine that's being developed for COVID-19, it's actually an RNA vaccine, so there's no chance of any type of infection or symptoms. Okay, so we talked in the last chapter about your innate immunity, and these were factors and mechanisms that you are born with and that are immediately available to you. But this is the immunity, your adaptive immunity or acquired immunity, that your immune system mounts only in response to the disease agent. So once you have an exposure, your immune system would mount this immune response against that particular disease agent. So it would be highly specific, but it would actually take a little bit of time to develop. And in the meantime, you would probably get sick. Okay, but like you probably have some experience with, either getting a cold or getting the flu and then having immunity to it. You actually oftentimes, not always, have memory against that particular agent, so that when you encounter it again, you might feel a little bit under the weather, but you probably wouldn't even know that you came into contact with that agent at that point, because you would mount such a rapid memory response, and it would be such a much stronger response that you would be able to fight that agent off when you encounter it for the second time. Okay, so our innate immunity was the focus of the last chapter, chapter 16, and now this acquired or adaptive immunity is our focus for chapter 17. Okay, so it's highly specific, and it's only going to develop once you've had an exposure. That exposure could either be a natural exposure or an artificial exposure. So a natural exposure would be like, say for instance, if you caught a cold and you mounted an immune response against the cold. An artificial exposure would be if, say for instance, you got a vaccine. Okay, so that's an artificial exposure. But in both of those cases, your active immunity, your immune system would be creating the response in response to either that natural or artificial exposure. A passive acquired immunity is when you are given ready-made antibodies. And so we've heard a lot in the news recently about COVID-19, the convalescent plasma, right? So that would be passive immunity. So where you're giving, and it would be artificial, where you're giving antibodies from a different person who has already been exposed and fought off the disease to a person who is currently fighting the virus for, and you're giving them those antibodies so that the antibodies can attack the virus and eliminate it, and presumably that person would be able to recover faster. So there is a passive natural transfer of antibodies, and that would be like the antibodies that are passed from a woman who has just given birth to the child through like the colostrum. Okay, that would be like a natural passive transfer of antibodies. On this next slide, I'm showing some examples of, like, for instance, your naturally acquired active immunity. Okay, that would be like if you caught a cold. Okay, but your body is still fighting it off, so that's your active immunity. Artificially acquired active immunity would be like when you get your flu shot. Okay, so it's artificially acquired because it's not a natural exposure, but it's still your active immunity where you are creating the immune response. A naturally acquired passive immunity would be the transfer of antibodies from mother to child across the placenta or in the colostrum. Artificially acquired would be like what I was talking about with the convalescent plasma from people who have had the coronavirus. Also, anti-venom for rattlesnakes, rattlesnake antivenom. That is another type of artificially acquired passive immunity. for that, you actually take the toxins, and they're inactivated, but you inject them into a different animal. Usually, it's a horse, and the horse creates an immune response against those inactivated toxins. So the horse doesn't respond like it's been bitten by a rattlesnake, but it has the toxins have been inactivated, but there's still proteins that can elicit an immune response. And so the horse would mount an immune response against those toxins, and then plasma. So blood would be drawn from the horse, and the plasma would be separated out, and the plasma would contain the antibodies, and then those antibodies could be used, like, say, for instance, if a person is bitten by a rattlesnake, you could give the person that antivenin, and the antibodies would bind to the toxins before the toxins could bind to your cells and cause a problem. So your natural immunity is going to take a couple of weeks to develop, and this was what I was talking about just a minute ago. So it only occurs in response to an exposure, and it takes a while to develop. And in the meantime, okay, you could get sick, but you're going to create a memory fraction, and that memory fraction could last a while. They're talking about, like, with the coronavirus, that they don't know how long that protective memory lasts. But if you think about, like, some of the vaccines that you got as a child, you're never boosted on those. So the memory actually lasts a lifetime. For others, like the TDAP, you're supposed to get boosted on it every 10 years. So those don't last a lifetime, but they last a pretty long time. But with the coronavirus, they're saying they're not quite sure how long the protective immunity may last. So this is kind of variable. With passive immunity, where you're just getting the antibodies, that is only going to last for as long as the antibodies themselves last. Okay, and antibodies in relation to other proteins are more stable than just your average protein because they have a lot of disulfide linkages. And so we'll see the structure of an antibody in a minute. So in general, they are very stable proteins, but okay, they're only going to last weeks to months. Okay, they're not going to last for a year. They're not going to last like your memory cells to an exposure would last. So they could last weeks, they could last a couple of months, a few months, but after that, for any type of immunity, you would need to rely on some type of active acquired immunity. All right, so I mentioned that the toxins in rattlesnake venom could create an immune response. I just wanted to talk about antigens for a second and what makes a good antigen. Antigens literally, like if you break the word down, mean antibody generating. So these are substances that are going to elicit an immune response. And the best antigens are large proteins. Okay, and the reason why the proteins need to be large is because the antibody is going to recognize a particular fold or a particular shape of the three-dimensional folded proteins. So as a protein gets smaller and smaller, and if you just had like a small peptide, a peptide might not be antigenic because it may not have enough secondary or tertiary structure. A good antigen could be a glycoprotein, okay, so a protein that has a carbohydrate attached. Some polysaccharides are antigenic as well, okay, but the best antigens would be large proteins, okay, and you can see in this figure some examples of antigens, okay, like for instance, the flagellum, okay, the flagellum is made of the individual proteins, globular proteins, flagellin, okay, remember arranged in that helical structure, the filament of the flagellum. Okay, so proteins, all right, that's going to be antigenic. The pilus, okay, or the fimbriae, those are made up of the protein pillin, all right, so again, those would be antigenic as well. Also the cell wall, all right, so the cell wall, the peptidoglycan is made up of peptido, right, protein, glycan, sugar, okay, so this is going to be antigenic as well. Okay, so all of these things that would be on the surface of the microbe, the pathogen, that would be exposed, okay, your body could actually elicit an immune response against those proteins. Okay, so say for instance, like in this figure right here, that is the protein flagellin. Okay, that's the protein flagellin. You could actually, on that folded protein, have several sites that could elicit an immune response, where you could have antibodies directed against several different sites on that particular protein. And those sites would be called epitopes, okay, or antigenic determinants. Okay, that's where the antibody is going to bind, all right? So the antibody would be raised in response to those particular folds on that large protein. Okay, so you we'll actually talk about the pregnancy tests, and for the pregnancy test, you need to have two different antibodies to that hormone, and for the test to be able to work. So there's an antibody against the alpha determinant and an antibody against the beta determinant. And that's how that test works, is that you need to have two antibodies raised against that particular antigen. Haptens are substances that are so small that by themselves they do not elicit an immune response. Okay, they're not antigenic, but when they are bound to a larger molecule, then they can elicit an immune response, because now they're part of this larger molecule, and the antibody can recognize them in the context of the larger molecule, okay, because they're just like one part or one shape of that larger molecule. So when we talk about your acquired immunity, we talk about your B cells and your T cells. And your B cells and your T cells are lymphocytes. And they originate in they're from your stem cells, which originate in your bone marrow. So your B lymphocytes are going to continue to develop and differentiate into mature B cells in your bone marrow, but your T cells are actually going to travel from your bone marrow, okay, via the blood to your thymus, okay, where they are going to develop and differentiate into mature T cells. Okay, so we'll see that your B cells and your T cells are going to carry out your immune response. And your immune response, your active immune or adaptive immune response, has a dual nature. So it can actually try to eliminate the pathogen before it infects cells, but once it infects cells, it can carry out a kind of a second type of attack, which would be to attack the infected cells by a cell-mediated response. So the first part of your immunity, which is carried out by antibodies, so the antibodies binding to viruses or the antibodies binding to bacteria, okay, kind of marking them for elimination, that's carried out by your B cells, and it's your B cells that create antibodies, okay, and this is what's referred to as your humoral immunity. Now, if that fails to ward off the pathogen and the virus actually infects the cells, or the bacteria infects the cells, then, okay, your T cells can carry out this cell-mediated response where they target the infected cells and kill them. Okay, so we'll see how your antibodies can opsonize, right? We talked about that with your innate immunity. They can mark a pathogen so that it is more easily recognized by phagocytes. And they are going to be most effective against pathogens when they are outside of cells. Once your cells are infected with a virus, remember how we talked about how, like, especially enveloped viruses would be expressing their glycoproteins on the surface of the infected cell, because when they bud from the surface of that cell, they would need to have their viral glycoprotein spikes in the envelope, in their envelope. So when your T cells can recognize these antigens that are like the viral glycoprotein spikes that are embedded in cell membranes, and carry out an immune response to attack those cells and kill them. Okay, so we'll talk about your cell-mediated immunity as well. It's important also to note, and we'll see this when we talk about T-dependent versus T-independent antigen, that your T cells also play a role in stimulating your B cells to produce antibody. So normally we associate B cells with your humoral immunity and T cells with your cell-mediated immunity, but your T cells, okay, specifically your T helper cells, also play a role in your humoral immunity. Okay, so as we talked about how your T cells develop in your thymus and your B cells develop in your bone marrow, and I talked about them differentiating. Okay, so they start out as stem cells, but then they differentiate into these highly specific cells that can recognize basically, any antigen out there. So they express antigen receptors. And so each B cell would express around 100,000 antigen receptors, and they would all be identical. But then between the different T cells, okay, every T cell is expressing an antigen receptor that is different, okay? And you have this vast array of B cells and T cells that are expressing all of these different antigen receptors that can recognize virtually any antigen out there, and that's how you're protected, okay, because you can mount an immune response against these pathogens once you encounter them. So you've got like all of these B cells and T cells, but based on your personal natural or artificial exposure, you might only activate a small fraction of those, okay? And everyone's immunity is different based on, where they've traveled and what exposures they've had. Okay, so you've got this vast array, and you probably won't use most of them, okay, but they're available to you, okay, and the antigen receptors, okay, are proteins, all right, and they have different shapes, okay, to bind to the particular shape of the antigen. And once they bind the antigen, okay, you're going to mount this immune response against it, okay, and that's what's referred to as clonal selection, and we'll go through that in just a minute. Okay, but your T cells and B cells, okay, they develop in the thymus and in the bone marrow, respectively. And as they develop, okay, they are expressing these antigen receptors. And once those antigen receptors are expressed, okay, on the surface of the cell, okay, then you have a differentiated B cell or T cell, and they can go out into your blood and your lymph and bind to antigen and mount an immune response against it. I mentioned it on this slide right here, 2,000 antibodies per second. Okay, and so they're very short-lived, all right? They only can, make this high output of antibody for a short amount of time. But your memory cells, on the other hand, okay, those are the ones that could last, from years to decades to even your entire lifetime, okay? And this would be what would be called the primary response, and we'll look at that in subsequent slides. This would be the primary response, and if your memory cells are activated, okay, you would go through an entire another round of clonal expansion where those memory cells would differentiate into two populations, the plasma cells and also another memory fraction. Actually, it would be a stronger memory fraction. And we'll see that, okay, when we look at the primary and secondary response. And that response would be faster, and you would produce a higher concentration or titer of antibody that would last longer. Okay, and that's when you may not even know that you've ever contracted the virus or the pathogen for the second time, because you have such a strong response that protects you. I just want to mention that, we talk about your lymphocytes, your B cells and your T cells being able to recognize virtually any antigen out there. And, so what if they actually recognize host proteins, right? Okay, so if they recognize host proteins, which would be called self antigens, okay, you don't want that to happen because then you would have your immune system would be attacking you, all right, which is what happens in autoimmune diseases. So if during development, okay, when you have your B cells and your T cells expressing their antigen receptors and differentiating, if they recognize one of your antigens, a self antigen, they go through a process which is referred to as clonal deletion. Okay, so clonal deletion, it's a self-induced apoptosis, okay, programmed cell death, where the lymphocyte is inactivated so that you won't have lymphocytes that are attacking you. Okay, this slide is looking at the anatomy of the antibody, and the antibody is actually pretty stable. And I mentioned that it's due to all of these disulfide linkages, right? So you can see on this figure where there are a bunch of disulfide bonds. Okay, the disulfide bonds are indicated in red. So, this antibody has quaternary structure. Okay, it's actually made of two identical larger proteins, okay, which would be called the heavy chains, and then two identical smaller proteins, okay, which would be called the light chains. And within the light chains and the heavy chains, you have disulfide linkages. And then between the light chains and the heavy chains, you have disulfide linkages, which are holding those polypeptides together to make the antibody protein. So you can see that the antibody has this Y-shaped structure, okay? And the stem of it, all right, is referred to as the constant region. And so for like an entire class of antibodies, right, we'll talk about the different classes in a minute, this constant region would be identical, okay, for the entire class. But, okay, it also has two variable regions, all right? And this is going to be highly different between the different antibodies, okay? And this is what is allowing each antibody on a B cell, okay, for the antigen receptor, which is basically almost identical to the antigen that is, the antibody that is secreted, allows its specificity. Okay, so in the variable region, okay, the shape is different, okay, and that's what allows it to bind to all of these different antigens, to be complementary to all of these different antigens. Okay, so the antibody has two variable regions, so it can actually bind to two antigens, okay, and that makes it bivalent, okay, so it can bind to two antigens. All right, so that variable region is actually the antigen binding site. So here are the different immunoglobulin classes, IgG, IgM, IgA, IgE, and IgD. IgG is the most common class of immunoglobulin, and it is a monomer. Okay, so you can see that it's just one antibody compared to IgM, okay, which you can see in the figure, actually has 5 antibodies held together by these proteins called J chains. When you think about like one of the functions of antibodies, which would be to agglutinate, okay, or clump together antigen, all right, for it to be disposed of, okay, or phagocytosed. The IgM is going to do that really well, right? Okay, it's going to be able to clump together antigen really well because it has 5 antibody, and each one of the 5 antibody has 2 binding sites, right? Okay, so it has 10 binding sites. So it's going to be the best at agglutination. Okay, so IgG is the most common. It's a monomer, okay? It is a good opsonin, all right? So it can coat a bacterium that has an antiphagocytic capsule so that a phagocyte can eliminate it. It can initiate the complement cascade. All right, so the classical complement cascade that is initiated by antigen binding to antibody. It can cross the placenta, okay, in passive immunity. We'll talk about this in a minute with the primary and the secondary response, but it is the major antibody produced in the secondary response. Okay, and it's also a good neutralization antibody, so it can bind to toxins and neutralize them before those toxins bind to your cells and cause a bad effect. IgM is secreted during the primary response. Okay, and we'll look at that in a minute. Okay, like I mentioned before, it's a good agglutinating antibody. It can actually activate complement as well. IgA has 2 forms. It can occur as a monomer, and it can also occur as a dimer. The dimer is the secretory form, and this antibody is actually found in your secretions. It is abundant in colostrum. Okay, so it's a really important antibody in natural passive immunity, and it is the idea with it being in the secretions is that it is to neutralize and bind to antigen before it can invade your cells. Okay, so these are the 3 most common, okay, in order. Okay, IgE would be, like I think makes up 80% of all of the antibody that you have. IgM would be the 2nd most common. IgA, the 3rd most common. IgD and IgE are a very, very small fraction of the immunoglobulin that you have. IgD is thought to play a role in that process that we just spoke about, clonal deletion. And IgE we'll talk more about when we talk about the hypersensitivity reactions. Okay, IgE actually does not play a protective role like the other antibodies that we're talking about in your acquired immunity. IgE, when it's bound to inflammatory cells like mast cells or basophils, can actually elicit an allergic response, which can cause the release of histamine, okay, which can actually, be quite damaging. Okay, so IgE is playing a role in your hypersensitivity reactions and is not part of your protective immunity. Okay, I think I'll stop right there, and this will be the first part of chapter 17.